PCR. PCR stands for Polymerase Chain Reaction. It was first reported by Mullis in 1985. PCR is a in vitro gene cloning. It is similar to replication. Only difference is that PCR is an in vitro process which synthesizes DNA, while DNA replication is the in vivo process which occurs naturally in our body of DNA synthesis. What is the principle behind PCR? The fundamental principle is to amplify DNA fragments, that is target DNA, into millions copies in a short period of time. Means we can get millions copy of DNA in a short period of time. This DNA template is amplified into 2 templates in first cycle and is amplified into 4 templates of DNA in second cycle of PCR. PCR machine can perform polymerase chain reaction. What are the requirements for PCR? We require template DNA, which is used for the amplification, and we require primer, which can bind to the DNA template, and dNTPs, which are DNA bases adenine, cytosine, guanine, and thymine, which can bind to the DNA template. For the extension, we require Taq polymerase, which is an enzyme isolated from the heat-stable species. It has capability to tolerate high temperature, and we require buffer for the pH maintain. Steps. There are 3 steps performed in PCR. First one is denaturation, second one is annealing, and third one is extension. Denaturation process can take 1 minute and at 94 degrees Celsius. In this process, the 2 strands of DNA are separated by the breaking of hydrogen bond between the 2 strands. Second process is annealing. It can perform for 45 seconds at 54 degrees Celsius. In this process, primer binds to the single strand DNA template. Third step is extension. It occurs for 2 minutes at 72 degrees Celsius. In this process, the nucleotides add to the strand and there is synthesis of new strand. What are the applications of PCR? There are various applications. I am going to tell you some few of the applications. First one is gene cloning and manipulation. Second one is diagnosis and monitoring of hereditary diseases. And third one is amplification of ancient DNA, etc. Thank you.